రెండు. This classification internal or endo. So external secretory is tissue and internal secretory tissue. Let's see the first one. Is it clear or not? Slide clear or not? Are you able to see the flow chart? Secretory systems, external, internal. Hello?
Okay, sir. Okay, sir.
Right. And again, actually, the tissue, two types, external, internal. And again internal, three types. These are granular trichomes, nectars, and idotodes. And again, nectars, floral and extrafloral. Internal, secretory cells, cavity duct, latticeous. In latticeous, first of all, in cavities, two types, lysogenous, cytogenous, latticeous, non-articulate and articulate. Let's discuss one by one. First, external secretory tissues. In the external first one, granular trichome. Let's see the granular trichome. So before that, let me give the brief introduction about external secretory tissues, which are occur external surface in the plants. They may be unicell or multicell. They may be made up of unicell part or multicell, with or without vascular elements. They usually develop from the epidermis. Most of these hairs develop from the epidermal region or subepidermal layer of the plant organ. Three main types of external secretory tissues are the glandular trichome, nectaries, hydatodes. These are the major three. And let's discuss one by one. Glandular trichomes. Let's see the first one, glandular trichome. If you see the glandular trichome, where it is, right. First of all, these glandular may developed from the epidermis or subepidermal region. Most of the epidermis region. They are unicell made up of single cell or multicell. Usually they have it is structurally it is two parts. One is stalk. You can see in this diagram. Are you able to see the diagram? Hello? Hello? In glandular trichomes, structurally made up of two parts, stalk and head. This is stalk. This part called as stalk, and the globostructure terminal part. This is called head. It's called head. The head portion will be secretory in nature. Actually, the secretory substances secreted by the head portion. The head's glandular trichomes are covered with cuticle, and which is the external covered with cuticle layer. The secretion accumulates in the space between the cell wall and the cuticle layer. For example, this is the cell wall layer, and which is covered by cuticle layer. If it is the glandular cell, so now this secretory substances, chemical substance deposited between cuticle layer and epidermis. So this is the chemical substances. The cuticle burst release the secretion. If the cuticle burst, automatically these chemicals release out. The examples of the glandular, you must have seen in our daily life, this type of glandulars. The most best examples are one is digestive glands in Drosera, salt gland of mangrove plants, collectors. First see the glandular. digestive glands. What is the function of digestive glands of Drosera? In Drosera, the capture digests to the small insect using the stalked mucilaginous glandula which are presented in the leaf surface. These mucilaginous glandula, it is called tentacles, which are present in leaf surface. These glands are glandular trichomes. This trichome Drosera possesses vascular elements. So what is the function of this glandular? As it is the slime nature, if insect trapped the leaf, the kills the release the digestive enzyme, especially proteases. This proteases digestive enzyme mostly the insect made up of proteinaceous. So which digests the protein, and these proteins converted into amino acids. From amino acids nitrogen received by plants. So this is the insectivorous plants. What is insectivorous plant? Insectivorous plants autotrophs or heterotroph? Hello. This insectivorous, what you see in this, you can observe the clear glandulars from the leaf surface, and this is the clear view of the glandular air. You can observe the both stalk and head region also. Right? Tell me, this is the best example in insectivorous plants. Now I would like to ask the simple question: These insectivorous autotrophs or heterotrophs? Hello, hello.
Heterotroph, sir.
What is heterotroph? What is heterotroph? What is autotroph?
Autotroph, they can prepare their own food. Heterotroph, they do not prepare their own food.
Now, is the diagram visible or not in this picture?
It is visible, sir.
The leaf structure, leaf color, which color?
Green color.
Which means,
Chlorophyll is present.
so chlorophyll present means is it autotrophs or heterotrophs?
Autotroph, sir.
So it is not a heterotroph. Remember, it is autotrophic plant. They can prepare their own food material. But why it is depend on insect? What is depend on insect? In which purpose? Halder told you from the insect they are mainly observe the proteins in the form of amino acids. So amino acid made up of amino acid. What is the which element present in amino acid?
Proteins.
Which element I am asking in amino acid? Which elements are present? Proteins, subunits, amino acids. But what are the elements in amino acids?
Lipids, sir.
Nitrogen. The only the purpose of these insectivorous plants kill the insect in the purpose of nitrogen. This type of plants majorly may grow in nitrogen deficiency land. You know the NPK, the major requirement to the plant growth and all plant activity, isn't it? NPK, nitrogen, phosphorus, potassium, they are the major role, may play a key role in plant metabolic activity. So, especially this type of plant, whether Nepenthes or Drosera, whatever it may be, this type of plants, any insectivorous plants, grow in nitrogen deficiency land. In deficiency land, there is lack of nitrogen. They have to depend on the insect for receiving the nitrogen from the insect. So it doesn't mean heterotroph. Remember it. Is it clear? So this is one of the glandular trichome. So next one, salt gland of mangrove plants. The salt glands also external glandular structure present on the epidermis of mangrove plants. Already told you most of these glandular hair from the epidermal region. They are abundantly present on leaves of the holophytic plants. The function of salt gland, The remove the excessive salt from the organs. As it present in the halophytes, that is saline water. In saline water, there is a chance to accumulate the salt in plant. What happen if the salt accumulated in the plant? What happen? Hello? If the excess of salt present inside the plant cell, what happen? The water moves outside. As a result, shrinkage happen. The plant would be die later. So to prevent this, the salt glandular remove the excess of the salt from these organs. You can see. Are you able to see this diagram? Salt crystal on leaf surface of black mangrove. Hello, hello.
Yes, sir.
Right. You can observe clearly in saline water. If the leaf receive excess of salt, the salt accumulate in the leaf region, but this excess of salt particles stored comes out and stored inside the glandulars. As a result, it is appear as a the crystal salt crystals on the surface of the leaf. You can see in this picture also. And another glandulars are collectors. The collectors are also specialized on more complex external secretory structure found in some plants such as Malus, Rosa, Pyrus. They are found on the young leaf of Primordia. The collector produce sticky fluid which cover the leaf Primordia. The collector drops off as the leaf Primordia matures. Thus, they provide protection dormant bud and young leaves, and also protect from other animals also. As it is sticky nature, the some insect trapped in this sticky nature. So this is the functions of this collector. And nectar is another specialized tissue. One is completed. One is completed. That is glandular in external secretory tissue. Glandular trichome completed. The glandular trichome presented in insectivorous example plant name Drosera, and another glidestive trichomes present in mangrove plants to remove excess of salt, and another type is collectors. And next type of external glandular tissue, external secretory tissues, nectar is. These also external structures, they secrete sugary fluid called nectar. Already you know the nectar, how the honey preparation from flowers. The nectar, the function attract the insects, thus helps in pollination. That is the first primary use. The nectaries are usually multicellular, majorly multicellular, but um, the first one glandular hairs may be unicell or multicell, but nectaries mostly they are the multicell. Can occur in the epidermis and hypodermis or in trichomes. Like a glandular, the nectar is also have the thick cuticle. The nectar accumulate in the cuticle and cell wall the same. The glandular substances also accumulate between the cuticle and the cell wall. The same here, the nectar chemical also accumulate between the cuticle and cell wall. During release nectar, the cuticle rupture. Then automatically the nectar secretes out. The two types of nectaries are present in plants, floral and extrafloral. This is the name clearly given. What is floral? Floral, they are common type of nectaries. They are associated with flowers, usually found in floral thalamus, base of the gynoecium. If you take the picture, example, let's draw diagram. The flower, I am drawing only one gynoecium part. Stigma style. So the next common anthers, next calyx, corilla, they are the common. They are the common thing. Usually found floral thalamus. So this is the where the ovary originated or positioned, situated. That is called thalamus. This is thalamus. So usually this floral on the floral thalamus, base of the gynoecium or androecium, base of the gynoecium or also base of the androecium. So the floral nectar is located at the base of the androecium ear or base of the gynoecium ear. From this nectars are released, the sugar substance nectars released out, right? From where? From where the insect receive the sugar substances. So the second one is extrafloral nectaries. What are the extrafloral? Without flower. Vegetative part such as petiole, pedicel, stem, they are the extrafloral nectaries. They are mainly found in Verbenaceae family, Bignoniaceae family, and Euphorbiaceae family. And the another important external one is idathodes. This is very, very important. Idathodes, external external secretory tissues are first one glandular, which is not an important one, but remember glandular. Second one is nectar is the third very, very important idathodes. It is important for short answer. Idathodes. Idathodes are the special type of secretory tissues in leaves. They are modified pores, especially on leaf, which exude water as drop. Actually, in generally, how the water loss, hello,
Yes, sir.
Sir.
in general, how the water loss from the plant body
Through stomata.
through stomata in the form of water droplets or in the form of vapor.
Vapor.
Vapor. That process called as
Transpiration.
transpiration. Actually, water loss through the stomata in the vapor position, this process called as transpiration. But some monocots plants, the excess of water lost through these idathodes. These idathodes actually specialized pores. They are not regular as stomata. Stomata subsidiary cells, stomata guard cells. They are different. The pores of idathodes also called as water stomata. They are different stomata such as called water stomata. They facilitate guttation. Guttation means secretion of water as droplet. Idathodes made up of the living cell called epitheme with numerous intercellular spaces. The cell of epitheme lack of chloroplast, vascular bundles of leaves, and the epitheme. If you see the diagrammatically, so this is the idathodes. These are the vascular tissue. Already told you the vascular tissue not end up to the tip of the leaf in monocots, especially grass family. What happens this grasses family monocots, the vascular tissue end until just before the tip of the leaf. Next, its continuation with help of the living parenchymatous epithelial cells. Later, this epithelial cell, let's take the marker. Epithelial cells end with substomatal chamber, where the water storage, excess water storage, through the water stomata, it is not a guard cell. There is no guard, actually guard cells present, but they are inactive state. There is no movement of the guard cells. There is no open and closing. In general, the transpiration process, the stomatal movement takes place. Stomatal movement, stomata open and close. When the stomata open, water loss. When the stomata close, water not loss. But here, it is not a activity of the guard cells. And actually, in general, and transpiration takes place in which time? High transpiration takes place in which time? Hello? Morning, afternoon, or evening? Hello?
Afternoon, sir.
Afternoon time. But here the guttation process mostly takes place in morning time. It does not depend on the sunrise. What happened during the morning time? The excess of water stored in the substomatal chamber. This excess in substomatal chamber released in the form of drops one by one. So this is by root pressure. Actually, the root pressure create in the root. This root pressure push the water from the root toward the upright direction until the water droplets released out. Generally, you must have observed in morning, early morning time, especially in winter season and also summer or winter, any season, while you walk on the grasses family, grass, while you walk the grass, what you observed?
Water droplets, sir. Water
Water droplets observed. And if the plants are very large, your the shirts, your clothes also will be wet. The reason is the guttation. Due to the guttation, water is released out. So this is about guttation. This is important. If you ask examination point of view, you should draw this diagram. Only this diagram, this one, not this. This is for your clear understanding. I will have to show this. But exam point of view, only this diagram enough. This droplets formation, no need to draw. But remember, this is the guttation. Droplet release, guttation. Vapor release, that is called transpiration. So in guttation, transpiration, mainly transpiration takes place in depend on light, but there is no dependence on light. There in transpiration, the active guard cells are there. Due to the activity, the stomatal movement, closing and opening, due to this water loss. But here there is no active guard cells. Instead of that, water stomata present. So this is the and also through the stomata, some minerals also loss. Minerals also loss. Apart from the water, including water, some minerals also loss, but not transpiration. Only water, but not minerals. So this is the some differences between transpiration and guttation. Right. Next one. So this is the short answer important. Remember. Now external secretary tissues are completed. Now internal secretary tissues. Internal secretary tissues is embedded inside the plant. They cannot be visualized externally. If we see glandular, we can observe. If we see nectar is, we can, even though we can't observe, we feel it. When you pick up a flower, when you, that mean, flower from the base, you feel as there some sweet substances. And iodotodes also we can see. Iodote water droplets. Internal secretory cells are there store secretory products. These are three types: internal and secretory cavities and ducts. Lattice for secretory cells, which are called idioblast, these are the specialized parenchyma cells. These are the specialized. Actually, this is made up of parenchyma cells and also called idioblast. The idioblast may be isodiametric, elongated, tube-like. There is the structure not important. Most of these cells contain secretory content. They can store tannin, mucilage, oil, crystals, and raw feeds, cystoliths. In the specialized tissue, idioblast cell, tannin present, mucilage, oils present, crystals present, raphides, cystoles also present. And secretory cavity or duct. These are two types, lysogenous cavity and cytogenous cavity. The lysogenous cavity, this cavity before secretory chemicals, group of cells, for example, this is the group of, in tissue, group of cells are there, like this. What happened during lysogenous formation cavity, in this group, some of the cells destroyed lysis, which is called as lysis. In this group, some of the cells here, some middle, this is the where I am using eraser. This is lysis occur. Due to lysis, what happened here? Cavity formed. After cavity formation, secrete some of the chemicals inside the cavity. It is not appear outside. That's why it is called as internal. So this is called lysis or lysogenous cavity. And next one, cytogenous cavity in the same way. In cytogenous cavity cells, the same one, two, three group of cells here also. But there is no lysis. But what happened here? These cells, this is intracellular space in middle. Let's change color. Middle, this is intracellular space. What happened in cytogenous? This intracellular space get extended. As a result, the surrounding cells are separated from each other, and as a result, this internal space become larger and larger. The finally it is settled as a large cavity formed, and the remaining cells as it is. That means the surrounded by common surrounding cells. So this cavity there is no lysis separate of the intercellular space increasing gradually. But here lysis occur. Here lysis occur. So that is bit different between lysogenous and cytogenous cavity. And example, lysogenous cavity, Rutaceae family, citrus and lemon. Best example, when you pick up the pericarp of the citrus, when you press, what you observe? Do you observe ever? Hello?
Glands, sir. White, brown structures.
Of course, brown structure. But what I'm asking, when you peeling up, when you peeling up pericarp from the fruit, when you press the pericarp, when you press the pericarp from the pericarp, what are released out?
Yeah.
When you press the pericarp, that means peeling.
Watery substances, sir.
Right, the strong smell water released out. So that is lysogenous, that is developed in lysogenous cavity. And cytogenous cavity examples, Apiaceae family, Ostraceae and Apiaceae family. Ostraceae and Apiaceae family. Do you know the Tridax procumbens? When when you cut the Tridax procumbens, some leaf like, the water like liquid comes exude out. So this is developed in cytogenous cavity. So you can see the images, lysogenous cavity formation and cytogenous cavity formation. What happened in lysogenous cavity? The all cells are lysis occur. Lysis in empty place. If you see cytogenous cavity, number 4, here the 4 number cavity, A gradual increasing. Gradual increasing. Example, pinus, resin canal. Example, pinus. And latissiferous latex, latissiferous tissues. These are look like a tube-like cells filled with fluid called latex. Milky substance latex. What is whenever you see the latex, they are the milky substances. Can you tell the examples of latex plant?
Jackfruit, sir.
And
Rubber.
rubber. But in our daily life plant, I am asking, can you
Jackfruit.
and the best example? When you cut the plant, the milk-like substances exuded. The milk-like substances called as
Supporter,
latex.
sir.
Right, sapota, calotropous, sapota, calotropous, and picas. They are all have the latex. And it is two type of latiferous in plants, non-articulate latiferous and articulate latiferous. In non, they are called latex cells. Single, they are not a group. They occupy the intercellular space of other cells. The mainly present at intercellular space, non-articular latiferous may be branched or unbranched. Of course, it is unicell. Maybe it branched or unbranched. Example, ficus, just now told you, the ficus. And articulate, this is now compound to that is not a cell type. It is accumulated cell, compound structure. The series of cells, those cells arranged end to end. Here, what happened in non-articulate latispheres? Here, the single cells, let me take marker. Single tube-like cells present single, maybe which is unbranched or like a branch like this, or branch. But articulated, it is compound. Compound means actually what happened? This is one articular cell, then with another latex cells, another cell. So these cells arranged end to end. The common wall between the cell may be interact or disorganized. And the common walls between the cells may be intact or disorganized. Articulate latispheres resemble to the xylem vessels. Already we completed the xylem vessels. Cylindrical, cylindrical structure. Xylem vessels are cylindrical tube-like structures. There are two types. Again, articulate latispheres, two types. Anstomizing and anastomizing. The anastomizing latispheres lateral connections are established with similar latispheres form reticulate, the same similar type. With its tubular, same connect with tubular. With its globular, same connect with globular. The best example, Hevia brevilensis, rubber, and Moni heart. Articulated, non-anastomizing. These are latiferous. No lateral connections are established. No connections. Hypomia, musa, convolvulus. So you can see the Hevia brevilensis, rubber formation, latex, white milk structure. And also from papaya also release the latex. You can see papaver somniferum. Papaver, what is the importance of papaver somniferum? Papaver somniferum latex. From papaver somniferum latex, drugs are prepared. These drugs affect on central nervous system. Cell structure, it is not important, but okay, anyway, the cells usually multinucleated, milky with latex, colorless medium, orange, yellow, papaver. So these are the different type of structures. What are the chemicals in latex? Many organic compounds present in this latex, such as organic acid, alkaloids, terpenes, resins, and enzymes. Latex of papaver somniferum contain alkaloid, morphine, codeine. Just now told you, morphine. Morphine used as a drug. Codeine. The latex carica papaya contain digestive enzyme papain. Latex evia contain rubber. The rubber particles are suspended in the latex. When latex is removed from the plant, the rubber particles clumps together. Right. So this is about secretory cells and tissues. Is it clear? Any doubt in this? Hello? Hello? Hello?
No, sir.
Right. This is very easy topic. That's why it's completed very fast. Next. And like what they given, I thought it's completed internal secretory tissue systems. Should I explain about tissue systems? Tissue systems, which means the group of tissues to form tissue system, epidermal tissue system, ground tissue system, vascular tissue system. Three types. How many types? Three types. Epidermal tissue system, number one. Next, ground tissue system, number two. And vascular tissue system. What are present in epidermal tissue system? Epidermal tissue system means epidermal complexes. What are the arranged from the epidermis? They are the epidermal substances, mainly cuticle present on the epidermis, and epidermal cell, costal cells, which are present in vein, stomatal complex. Do you know stomata complex? What is present in stomata complex? Already, already you know already you must have learnt in during practical training classes. Different type of stomatal type. First, tell me the stomata structure. In general stomata structure. Hello. General stomata.
The
Guard
guard
cells.
cells are present, sir.
Right, guard cells. And next guard cells surrounded by.
Subsidiary cells.
Right, that's it. Subsidiary cells. So this is called stomata complex. This is called stomata complex. And what they given here, stomata complex types, I think, types given. Distribution of stomata. Distribution of stomata, amphistomatic, hypostomatic, epistomatic. What is amphistomatic? If you take the leaf, for example, this leaf internal structure, upper epidermis, lower epidermis, right? Where are the epidermis? Sorry, where are the stomata present? Hello? Stomata region. Location of
Generally on
stomata.
the lower epidermis, sir.
In epidermis, leaf epidermis, mainly majority found in leaf epidermis. Generally, the normal plants amphistomatic. Amphistomatic means let's change color, blue color. Here the stomata, upper epidermis, stomata, upper epidermis as well as lower epidermis. In both epidermis, stomata present. That is called amphistomatic. And the next one, hypostomatic only lower stomata, lower epidermis. The same if you take the epidermis, upper epidermis, lower epidermis, but stomata present at lower side here only, but not upper. This is hypo. Next, epistomatic. The stomata, at the upper epidermis. The same. Only upper epidermis. This is epistomatic, mainly present in water plants. Now, most of the plants are xerophytic. This type hypostomata present, right? After this is called distribution. If asked about stomatal complex, you write about stomata structure, stomata distribution, and stomata types. Types also, parasitic stomata, diacytic stomata, anomocytic, anesthetic. What is anomocytic? Hello? Hello?
Yes, sir.
Tell me anomocytic type. In anomocytic, what happened here? Let me change color. Guard cells, on the guard cell, three subsidiary cells of one, which one is smaller. This is small. Are larger than the other two. Are larger than other two. One is smaller, remaining are larger. One is larger. One is medium. Like this. So this is anomocytic. Anomocytic means irregular. There is irregular shape. That is called anomocytic. What is parasitic? Parasitic. Parasitic means, for example, this is guard cell. This is guard cell. The subsidiary cells are parallel to the guard cell like this. Subsidiary cells to parallel. That is parasitic. What is diacytic? Hello? Stomata. Rectangular to the stomata like this. Subsidiary cells like this. Right? So these are the types of the stomata. Distribution clear, types also clear. And any other? So this is all in epidermal. This is completely belongs to the epidermal tissue system. And what is next? What are given in your book? A trichome complex. Just now we discussed about trichomes. Maybe the trichomes unicell or multicell. The trichomes also emerged from the epidermis. Same, it has the stalk and head. But here the different type of trichomes, unicellular trichome, uniseriate filiform trichome, uniseriate macroform, biseriate trichome, multiseriate trichome. The name itself, the word name itself given. Unicell, it's made up of unicell. Uniseriate, only the series of cells arranged one above the another. Biseriate, two type of series. That's it. This is about the epidermal tissue system. And another one is fundamental tissue system and ground tissue system. What are present in fundamental tissue system and ground tissue system in internally? So if we take the internal structure, whether it is root or stem or leaf, the clearly epidermal parts, whatever it may be, hairs, trichomes, stomata, they are all completed. And what are the fundamental tissue? In orderly, that means what is next after? What is next after the epidermis? Hello?
Hypodermis.
Right, hypodermis. Next,
Cortex.
cortex. Just wait. Let me take a slide. Fundamental tissue systems. Is it visible? Fundamental tissue system visible or not?
Yes, sir.
Right. A fundamental tissue system or another name is called as ground tissue system. In this mainly present in the cortex, whatever you said, that is hypodermis and normal cortex, and these are the part of the cortex. cortex, endodermis, pericycle, pith. This is all fundamental tissue. So what is the other internal part apart from this? One is epidermis completed. Next, all are completed. Cortex completed, endodermis complete, pericycle complete, and medulla, pith complete. What is remaining in center position? What is remaining in steel?
Vascular bundles.
Right, vascular bundle tissue system remaining. I will explain later about vascular bundle system. Now let me complete fundamental tissue system. As it is, I will wind up very quickly. Cortex tissue, first of all, in ground tissue, first cortex. Just it is beneath the epidermis, which surround the central cylinders called cortex. It is delimited from vascular cylinder by endodermis. Right, by endodermis, vascular cylinder, all steel. Now, usually this cortex stem consists involved parenchyma with intercellular space, and also not only parenchyma and also collenchyma also, and chlorenchymatous stem. The cortex cells also contain starch, tannin, crystal, other common secretions. The cortex may contain here given collenchyma made up of sclerenchyma, sclerites in addition to parenchyma. Collenchyma could be arranged. What you said just hypodermis. Collenchyma mainly present in hypodermis, which is just beneath the epidermis. In dicot, collenchyma is often seen ridges in corner and in other position give temporary support to the plant. The function of collenchyma give support. Collenchyma develop just below the epidermis, forming the outer protective layer. Hypodermis, just you said. The inner layer cortex, endodermis. Between the hypodermis and endodermis, there is normal cortex. So cortex region usually consists parenchyma, not only root and also shoot, also stem and leaf. The mainly cortex made up of parenchyma cells, but not hypodermis. Function of cortex, if you see the cortex, it is protective tissue as it consisting the hypodermis and carbon assimilation as it in middle of cortex. You have the chlorenchyma cells. Chlorenchyma, what is the function of chlorenchyma? Hello?
Yes, sir.
Chlorenchyma function. What is chlorenchyma first?
It is a parenchymatous tissue, sir, which helps
Okay.
in perform in photosynthesis.
Why it is perform photosynthesis?
Presence of chlorophyll.
That's
Chlorophyll
it.
is present in.
That's it. Which parenchyma consists the chlorophyll? These are called chlorenchyma. That's also this type of cortex perform photosynthesis by carbon assimilation and also in water plant storage in xerophyte storage of water, and also it is functions as storage of food, help in temporary mechanical support to plant body, storage tissue help in pumping of water from root hair to the xylem. Just now we discussed about this guttation. It is also involved in guttation. And this is about cortex. Actually, cortex end with one single layer that is called endodermis. You can see in diagram also red color line. In diagram, this is epidermis. This is very important in internal structure, root internal, shoot internal. That's why I will explain here. I am explaining here. This is epidermis. This layer, this is complete cortex. This is endodermis. Also known as starch sheath in stem due to the presence of starch. How many layers it is? Uniseriate. Like a epidermal cell, it is also uniseriate. It is delimiting the cortex from the steel. Consists of barrel-shaped cell without internal space. In endodermis cells, in epidermis also no internal space. Endodermis no internal space, only internal space in cortex. The cells endodermis are living. The stem it is inconspicuous and wavy. In roots, it is well defined. Of course, this endodermis present in stem and also root, but it is not appeared in leaf internal. Majorly found in root. These are two types of endodermis. Hello, who is this? Zoom user, don't draw. So this is about endodermis. It is two types: primary thin-walled and secondary thick-walled. The primary thin-walled cells certain thickening suberins are developed in the form of bond strip, which is called as a Casperin bonds. In primary thin-walled, certain chemical thickenings by the suberin developed and form the bond or stripe, which is completely around the cell radial walls, and it is called as Casperin bond. The secondary thick-walled endodermis cells and the cell walls are thick by suberin. That mean actually, so this is actual endodermis. In endodermis, where the Casperin bonds are present here, like this, the thick-walled suberin chemical present. These thick-walled chemical suberins called as Casparian bonds. Among the thick-walled and walled endodermis, many roots, they occur occasionally thin-walled cells opposite to the protoxylem. These are called transfusion cells or passage cells. Transfusional cells pass through this passage are right here given passage cells. Through the passage cells, the sap absorbed by root air enter the xylem elements. Here, what happened here? For example, here, this endodermis cells. It is thin-walled. This thick-walled layer suberin with the male of suberin. These suberin chemicals, which is called as Casparian bonds. But here there is no Casparian bonds. These are called passage cells. Opposite the passage cells, xylem cells are formed. Here, xylem cells. So the
Sir,
passage cell,
passage,
yes,
passage cells and transfusion cells both are same, sir.
same. Both are
Okay.
same,
Okay.
right? Through the passage of transfusion cells, water moves toward the xylem, root xylem. From root xylem, it goes towards. This is the connection: root xylem to shoot xylem to leaf xylem. So water moves from the root to the shoot and shoot to the leaf, and hello, finally water goes
To the leaves.
from leaf xylem to leaf xylem to. Ayo, the water deposited in xylem, leaf xylem, the entire water deposited in leaf xylem. Is it possible? From the leaf xylem, the water
Not the,
moves
to out of the leaf.
out. How goes to out of the leaf?
Through stomata.
Right, that's it. Actually, this leaf xylem to mesophyll cell, to mesophyll, leaf mesophyll cell to the epidermal cell. So the stomata opening, when the stomata opens, the leaves evaporate. So water evaporate. This process called a transpiration. Right. So this is about xylem cells. Sorry, endodermis cells. Next, function of endodermis simple, protective, root pressure maintains root pressure. All I told you, due to the root pressure, guttation formed act as an air dam which prevent the diffusion into air to serve as storage tissue having the starch grain, many dicots. Dicot, only dicot stem mainly dicot stem. And now another ground tissue pericycle. Where is pericycle?
Below the endodermis.
Right below the endodermis and the start point of the steel. In the steel starting layer, pericycle. In dicots, pericycle is a multi-layered between endodermis and vascular bundle. Called vascular bundle not completed. It always occurs as a thin cylinder of tissue completely encircling the vascular bundles in width, that which means simple. Let me take pen. Where give any diagram? Not given. Okay. For example, this is steel. This layer, the steel limiting layer. This is pericycle, center medulla or pith, center medulla or pith. Now, between what is present in between pericycle and pith?
Xylem and phloem, sir.
Right, xylem and phloem together called as vascular bundle, VB, vascular bundle. Let me write in short name, vascular bundle. Right. The pericycle consists parenchyma cells, but not always. Mostly parenchyma and also maybe which are made up of sclerenchyma and collenchyma also. It may be a complete sclerenchymatous zone, consists of both sclerenchyma and parenchymatous, endodermis altogether absent. When endodermis altogether absent, the pericycle merges with cortex, right? If you take some some plants, there is no endodermis. Actually, what is the endodermis? Endodermis is the limiting between cortex and steel. If there is no endodermis, the endodermis replaced by what is the next layer of endodermis?
Pericycle.
That's it. And in root from the pericycle, lateral roots are raised. Roots of certain angiosperm parasites and aquatic plants lack of pericycle. Why parasitic plants and aquatic plants no pericycle? What is the reason? In root internal structure, pericycle function, lateral roots arise the lateral root from the pericycle. For this, the pericycle absent in the parasitic and aquatic plants. What is the reason? Why the parasites and aquatic plant no pericycle? What might be the reason? Hello?
In aquatic, the root system is not well developed. Maybe.
Okay, right. Next in parasites. Same, because parasites also depend on other plant. That's why is there any requirement of the roots?
No, sir.
Yeah, the main function of the pericycle formation lateral roots. From lateral roots, sublateral roots are formed. Rootlets are formed. The function of all these absorb the water from the underground in soil particles. In hydrophyte, actually, the hydrophyte itself present found in the water. There is no special roots to absorb the water because the almost all plant pots receive water, and also parasite as it depend on the other plants from the other plant directly receive the water, minerals and food. That's why both there is no pericycle, there is no lateral root system. Of course, no root system. In parasitic plants depend on other plants. It not depend on land. It not attached to land. Right? The
Okay, sir.
what functions in dicot root cells, pericycle become meristematic and form the cork cambium. Already told you the sometime where permanent tissues can metamorphose into meristematic tissues. So actually the pericycle is one of the permanent tissue. But this pericycle in dicot root can change into meristematic tissue and form the cork cambium. I will explain it during secondary growth. The pericycle gives rise to lateral roots. In stem, adventitious roots originate from the pericycle, where the adventitious roots are arranged. Where the adventitious roots, give me one example plant.
Grass.
Grass, fibrous roots. Of course, some grass adventitious also emerge. GMS, maize adventitious roots from the stem. Banyan tree, adventitious roots there. And pith, it is also called medulla, center, region, stem and root. Usually the pith dicot stem is largely parenchymatous. No, there is no chlorophyll, but leucoplasts are present in it. No chlorophyll, remember, only chlorophyll present in cortex region. No chlorophyll pericycle, no chlorophyll endodermis, no medulla. Intercellular space is seen in pith. What about pericycle? Pericycle intercellular space present or absent?
Absent, sir.
Absent. Like endodermis, no intercellular space in pericycle also. Pith, it is made up of several layers or intercellular space present. Uh, intercellular space is seen. In monocot stem, the vascular bundles are found and scattered throughout the ground tissue, and the pith is not distinguishable. Do you know monocot stem? If you see the dicot stem, the clearly differentiated pith region, vascular bundle as the ring. But in monocot, complete ground tissue, we cannot find out even where the cortex, where the endodermis, where the pericycle, where the medulla. In dicot root with is scanty or lacking, but monocot root is well developed. In dicot root with very less, monocot well developed. Actually with parenchymatous, but in some monocots, this medulla made up of sclerenchyma. From the medulla, medullary rays are arranged, or pith rays called as pith rays. The medulla, certain plant is partially obliterated during the growth of the stem, and such cases stem becomes hollow. That means, do you know ricinus stem? Hello, a ricinus
Yes.
stem and some water plants, Nymphia nilambo, petiole. If you press, if you press this type of stem, the easily it gives the tough sound because of actually the pits at what happened in actual internal center in central. Just wait. epidermis, cortex, endodermis, steel. For example, in center, this is actually completely occupied by medulla. But in this area, there is no medulla. It is completely formed as a empty space, like hollow structure, like empty space, empty space. If you see one side, you can observe other side also. So, however, in such cases, nodes retain their pith. Certain pith cells possess tannin and crystals. These are the chemicals, tannin and crystals also present inside the medulla. Right, next one. Function of pith storage. If it is made up of sclerenchymatous, it is gives mechanical support, and medullary rays serve as a channel from transport of food, lateral movement of water. In dicot, some of the parenchyma cells, the medullary rays become meristematic and gives rise to inter. This is important. In dicot, from parenchyma cells of the medullary rays, these medullary rays arise from the medulla. The function of this medullary rays can perform as a meristematic tissue and form cambium. I will explain in secondary growth. Right, this is about ground tissue. And next one, intercalary tissue. Let's see intercalary tissue. Sorry, vascular tissue system. Vascular tissue system. Vascular tissue system also important. Are you able to see vascular tissue system? Hello?
Yes,
Yes,
sir.
sir.
The vascular tissue system also short answer important and very easy. And the vascular tissue system consists of number of vascular bundles which are found to be distributed in the steel. Already have drawn the picture. Center medulla, outer pericycle, and in between vascular bundles. In the steel is the central cylinder portion of the stem, and the root commonly surrounded by endodermis common and consists vascular bundle, pericycle, pith, and medullary rays. These are all explained except the vascular bundle. In root separate xylem and phloem strands are found. For example, this root, this is phloem stand, xylem stand, phloem stand, xylem stand, phloem. This is called radial arrangement. If you take the stem, if you take the stem, in the stem, uh, the vascular bundle arranged in ring like this. These are the vascular bundle. Are you able to see? Hello?
Yes, sir.
Right. This vascular bundle in C. What is present in between here? This gap filled by?
Pitta.
Are you able to? Right. Function of vascular tissue system already we discussed last class. Transport water, minerals. No need to waste your time. Also, procambium also we discussed. Where is procambium first? Already it is discussed. Actually, from the procambium, these vascular bundles elements are derived. Vascular bundle elements means xylem and phloem elements. Xylem and phloem elements derived from the procambium region. Vascular bundles, the major two zones, xylem or wood, phloem, bast, cambium. Xylem is already completed. Xylem completed by trachea vessels, wood parenchyma, xylem parenchyma, secondary wall thickening. These are all completed. Was it complete or not? Hello? Last class
Yes,
about
sir.
phloem, the phloem elements also sieve tubes, campion, phloem parenchyma, cambium, types of vascular bundle. Now, the radial, conjoint, concentric. First, let's see radial. Radial vascular bundle present in root system. In radial, for example, this is the steel. In steel, the arrangement of xylem and phloem radially arranged. Here xylem, alternate phloem, and then again xylem, alternate phloem, xylem. So this type called as radial, which is mainly
Radial.
present in roots, which are mainly present in roots of angiosperms. Conjoint vascular bundle. In conjoint, closed and open type. What is conjoint type? Xylem and phloem together form a bundles. Two subtype collateral. These are one is collateral. In collateral, xylem and phloem lie together in the same radius, which means vascular bundle, xylem, phloem. Where it is xylem and phloem, same radius, but not different. If it is different, that is radial. So same radius. Same radius, xylem inward. Where is xylem? Xylem inward. Here, phloem outward. Here, phloem occurs one side of the xylem stand. In dicot stem, cambium is present between the xylem. Let's change color. Cambium. This is cambium. Cambium present between the. This is very, very important for secondary growth. Listen carefully. Cambium present between the xylem and phloem. Now it is collateral open vascular bundle. If the cambium present, that is open type, open vascular bundle. What happened in monocots? In monocots, this cambium is absent in between xylem cell. Same arrangement, same radius, but no xylem. Sorry, no cambium. There is no cambium. If there is no cambium, that is called closed type. Present open type. Phloem collateral closed vascular bundle, monocot stem. Best example is monocot stem, right? And concentric, another one is concentric. Here given clear given diagram, open vascular bundle with cambium, without cambium, closed vascular bundle. Are you able to see the diagram?
Yes,
Hello?
sir.
Now bicollateral vascular bundle. Uh, Bicollateral means the phloem is found to be present both side of the xylem. It is present xylem center, outer phloem, inner phloem. That is called bicollateral. Present in Asclepidaceae family, some Cucurbitaceae family. Concentric vascular bundle. These are the primitive type. In concentric, mainly present in fern, Pteridophyta, some amphi, some of the drusinal plants also. Let's see the concentric amphicribal amphibasal. What is difference between amphicribal and amphibasal? Can you tell? Difference between anticribal and amphibasal. What you observe in this both
Xylem and
diagram?
phloem difference is there, sir. In amphicribal, phloem is outside, xylem is inside.
That's it. So the vascular tissue is also completed. Now the secondary growth. Important one is secondary growth. Let's wait 1 minute. Right. secondary growth. Are you able to see the diagram, PPT?
Yes, sir.
This is very, very important for short, for long answer, and also and important in anomalous secondary growth. If you develop concept about the secondary growth, the anomalous secondary growth will be easy to you. Already you must have learned anomalous secondary growth in during practical training classes. Are you learned or not?
Yes,
In
sir.
in paper 3, Boheravia stem, Dracena stem, Bignonia stem,
Yes, sir.
Amarantha stem. Right. What is secondary growth first? What is secondary growth and what is primary growth? How primary growth occurs? How secondary growth occurs? Already told you when I explained tissues. Tell me primary growth and secondary growth and importance of both. Already clearly visible in slide. Increase
Sir, in
diameter.
primary growth, in secondary growth, sir, the plant will show the woody structure and all the cork and all will be developed. The thickness of
Right.
the plant will be developed in secondary growth.
The simply diameter thickness increasing. In primary growth only the upright direction grow. What happened during the primary growth while the formation of branches? If the only the plant shows primary growth, can bear the developing branches, the thin stem can support the branches. If the main stem is very thin, only the plant shows primary growth without secondary growth. The plant can stand.
No, no, sir.
No, because apart from the primary growth, the secondary growth also required. Now the secondary growth increase diameter of the stem. In woody plant, the secondary tissues continues the bulk of the plant and it part of the providing protection, support and conduction of water and nutrient. After formation secondary tissues, providing protection, how it is providing protection? Protection, as you said, after formation bark give protection. After formation secondary tissues, conduction of water and food material. The secondary tissues are formed two types of lateral meristems, vascular cambium and carp cambium and phellogen. I will explain clearly one by one. The vascular cambium produces secondary vascular tissues while the phellogen form the periderm. So here the first one. In secondary growth occurs in perennial, no need. The formation of secondary layers tissues, right? Is there any diagram? Okay, not given, right? Simple, actually you don't know phellogen, phellum and bark, how the formation bark, you don't know. First, the formation of secondary vascular tissue. Remember, first of all, the secondary growth two types, intrastellar and extrastellar. First, let me complete intrastellar. In intrastellar, let me draw diagram where with the khali place, empty place here. Okay, let me take marker. This is primary plant structure. For example, this is steel. I have taken only steel. In primary structure, steel with vascular bundle. For example, this is the vascular bundle. What is present inside the sir? What is present in vascular bundle? Hello?
Xylem and phloem, sir.
In between xylem and phloem? What is present in between xylem and phloem?
Cambium. Cambium.
Right. First, let me draw cambium. Cambium. Yellow color line. Cambium. 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 And after that, inside, inner side, xylem. Outer side, phloem.
Phloem.
Right. So this is primary structure, only cambium present. Now this cambium, that means inside the vascular bundle cambium, that is called as intravascular cambium. That is called as intravascular cambium. Right. Let me write here. Vascular cambium, or it is called as vascular cambium. This one, vascular cambium. Vascular cambium. And what are present between? What is the present in between the vascular bundle? This place.
Pith,
This
sir.
place. Right. It is pith or medulla, the green color one. What are arranged from the pith or medulla? In between vascular bundle, what are present?
Transfusion tissues, sir.
Rays, medullary rays in between. Actually, medullary rays are present like this. Medullary rays. Let me change color. Medullary rays erased from the pith region. Already told you when I explained about the medulla, the function of medullary rays. What is the function of medullary rays? Medullary rays can perform as a meristematic tissue, right? This medullary rays. Now next. So actually this medullary rays develops as a cambial cells, meristematic cells. They actually these rays are not a meristematic cells. They are permanent cells. Already told you whenever necessary, they can perform as a meristematic cells. Now these rays can turn into meristematic cells and developed as a cambium. Now developed as a cambium. Now the where are the cambium? How many types of cambium? Inside the vascular bundle and outside the vascular bundle. Okay, cambium ring. This is called as a interfascicular cambium. This one. This cambium. This cambium. Interfascicular. Oh, already time 3 o'clock. Sorry. I will explain tomorrow about the secondary growth. I have a meeting. Is it clear so far? Hello?
Yes, sir.
Yes, sir.
Right. If any doubt, ask tomorrow. I will explain.
Okay, sir. Thank you.
Thank you all.